Hi folks, welcome to lesson 3 of unit 1 of the advanced higher biology course. Today we're going to be looking at membrane proteins and we're going to be continuing to learn more about the proteins that we learned about in 1.2, but we'll be applying it strictly to the proteins that we find within the cell membrane. Okay. Now, we already have some knowledge of the cell membrane. We know from National 5 that it consists of phospholipids and proteins. But what we're going to do today is we're going to learn a little bit more about the variety and the different functions of the proteins within the cell membrane. So there is a way of displaying the cell membrane, which is through something called the fluid mosaic model. Now, I've made this cell membrane image here, and it's probably not quite as good as some of the ones you'll see online. If you have a wee quick search for fluid mosaic model of the cell membrane, you'll see it has more of a sort of fluid shape. It sort of waves up and down, back and forth, sort of like, like if you watch the water frothing up and down, with all those waves in it. That's more like what the fluid mosaic model looks like. Mine is a bit more structured and two-dimensional, but there was a reason I did this to show you specific parts. Okay, so do look up the fluid mosaic model. It actually comes from the old mosaics that they used to make back in Roman times. They would take little tiny tiles and they would lay them in a certain arrangement so that each of the individual tiles didn't show much particularly, but the overall arrangement showed you the full picture. So that's where the fluid mosaic model comes from, because the cell membrane is not just it's not just monochrome. It has got the phospholipids and the proteins, but it's got a whole bunch of other molecules on there as well, which give it that sort of mosaic look. So we don't need to know about all of those other molecules particularly for this part of the topic, but we do need to know a lot about our proteins, okay? Now, we know that from our study of proteins that the amino acids, the building blocks or the monomers of proteins, they have R groups in them. Now, proteins within the cell membrane remain within the cell membrane, okay? Things like this integral protein here. Now, that gets to remain within the cell membrane because it has lots of regions of hydrophobic R groups. Hydrophobic R groups. And if we remember, hydrophobic means water hating, or it doesn't like to be near the water, okay? And we've got water or liquid on either side of the membrane. So therefore, these proteins like to be connected to the center of the membrane. And that's what holds them in place here. Lots of little hydrophobic interactions here and over here as well, okay? And that's what holds these integral proteins. These ones that are in the internal part of the membrane there are held together by these hydrophobic R groups. Now, we've also got a phospholipid bilayer, okay? That's two layers of phospholipids, and they act as a barrier to ions and most uncharged polar molecules, okay? So ions and uncharged polar molecules cannot pass through the phospholipid bilayer. They have to use one of these channels instead, and we'll talk more about the channels later on. Okay, so the phospholipid bilayer is a barrier to most ions and most uncharged polar molecules. Okay, so you need to remember the role of the phospholipid bilayer there and how the integral proteins are held within the membrane, and that's through these regions of hydrophobic R groups. Now, some of these integral proteins are uh, transmembrane proteins, which means that they span from one side of the membrane to the other. Trans, we know in biology, means to cross. So it crosses from one side of the membrane to the other. We can see one side of the protein here on this side of the membrane, and the other side of the protein over on the opposite side of the membrane. So we can see this integral protein here is a transmembrane protein. This integral protein here is not transmembrane because it doesn't span from one side of the membrane to the other. Okay, it's still an integral protein because it's connected through its hydrophobic interactions here to the center of these phospholipid bilayer, but it is not a transmembrane protein. Okay, it's integral but not transmembrane. This one here is integral and also transmembrane, and so is this one over here. Okay, now the integral membrane proteins interact extensively with that hydrophobic region of the membrane phospholipids, okay? So here, and here, and here, and here, and here, where those little red dots are, okay? We'll see an extensive interaction between those proteins and the hydrophobic region there, and that'll be due to the hydrophobic R groups that are located on them. Now, peripheral membrane proteins, such as this one here, and this one over here, okay, this one here, and this one here, we can see are located on the periphery or the outside or the surface of the cell membrane. And the reason that they're located there is because they have hydrophilic R groups, okay? So those ones like to be on the exterior, and the hydrophilic head of the phospholipid bilayer, okay? And they are bound to the surface of these membranes, mainly by ionic and hydrogen bond interactions, okay? Ionic between our basic and acidic R groups that we learned about in the last topic. Now, many of our peripheral membrane proteins interact with the surfaces of our integral membrane proteins. And we can see an example of that here. We've got a peripheral protein here, and it connects with this integral protein here. Okay, many of the peripheral proteins interact with the integral proteins here. We can see another peripheral protein which doesn't do that. There are some that don't, but this one here does interact with the integral protein, and we need to remember that. Now, molecules can move across the membrane. We know this from all the way back in National 5, where we learned about diffusion, osmosis for water, and active transport. And just a quick recap of those processes, remember, diffusion is the movement of molecules from a higher concentration to a lower concentration across the cell membrane, and the active transport is the opposite. It goes from a lower concentration on one side of the membrane to a higher concentration on the opposite side through the membrane proteins. Okay, and that will become important as we go on through this topic. So some small molecules, such as oxygen and carbon dioxide, pass through the bilayer by simple diffusion. Okay, we probably already knew that from National 5. Oxygen, carbon dioxide, and other small molecules will pass through the bilayer by simple diffusion. We can see that in the Amoeba Sisters GIF here. Now, there's another type of diffusion which we need to know about at advanced higher called facilitated diffusion. Now, that's the passive transport of substances across the membrane through specific transmembrane proteins. Okay. Now, note that word specific there because that's important. Now, we said earlier on our transmembrane proteins are these ones here, which span one side of the membrane to the opposite side. So when we're using facilitated diffusion, the molecules can't just pass through the phospholipid bilayer. They have to go through this protein, okay, this transmembrane protein. And the word there, specific, is important because only specific molecules can pass through specific transmembrane proteins. There are specific channels for each molecule or ion that looks to pass through these transmembrane proteins. So to perform specialized functions, different cell types have different channel and transporter proteins, okay? So it depends on the cell type, but each of them will have different channel and transporter proteins depending on the molecules or ions that they have to transport in and out of their particular cell. Now, most channel proteins in an animal and plant cell are highly selective, okay? And that comes back to the specificity we talked about up here, where they're highly selective because only certain molecules or ions are able to pass through that particular channel protein. Okay, so we can see in this GIF here, we can see various channel proteins, and you can see they're each color coded to indicate that they are letting different molecules or ions pass through their channel. So I'm guessing that maybe this red and blue molecule here can only pass through that channel. This yellow one there can only pass through that red channel, and this green molecule here can only pass through that green channel. The green one couldn't pass through here, the yellow one couldn't pass through here or here, and the red and blue one couldn't pass through here or here. Okay, so these molecules are highly specific to a particular channel protein, and that obviously depends on the particular cell it's in, and they're very selective, which means they only allow their specific molecules to pass through. So these channels, what are they actually composed of? What is their structure? Well, they're multi-subunit proteins. Okay, we learned about these in the last topic, multi-subunit proteins, with the subunits arranged usually at the sides here, okay, 1, 2, 3, 4, for example, in these corners. And then we have this channel which goes through the middle, and this is a water-filled pore, okay, and it extends from one end of the membrane to the other. So they have this channel or pore that's full of water, and that will allow the molecules to pass through this channel protein. Okay, so that's the structure of these channel proteins. Now, some of our channel proteins are what we call gated, okay? And you can probably guess by the term what that means. It means that they are closed at times and open at other times. Okay, so some channel proteins are gated. And what they do is they will change conformation. Remember, conformation is the shape of the protein. So they'll change conformation to either allow or prevent diffusion from happening. Okay, and there'll be different reasons why the cell would want to stop diffusion or allow it to happen, okay, depending on the particular circumstances of the cell and what it needs to do at that particular time. But we just need to know there are gates that change conformation to allow or prevent the diffusion of that. And we can see two examples here in our GIFs there. Now, there are there are two types. There are ligand gated channels, and as you can probably guess from the name, they are controlled by the binding of signal molecules, okay, i.e., ligands. All right, so the binding of these signal molecules will either open or close the channel. We can see in this top GIF here, it's the binding of these ligands to the protein that cause a conformation change that opens the channel. We also have to know about a second type called voltage-gated channels. Now, again, as you could probably guess by the name, voltage-gated channels are controlled by changes in ion concentration. And we can see here in this GIF as well, we've got quite a high, much higher concentration of positively charged ions on this side of the membrane than we have on the opposite side. And so when it obviously reaches a particular concentration for this particular voltage gated channel, it will open and allow the flow of these positively charged ions through to help balance what we call the membrane potential, the electrical difference in terms of the charge on either side of the membrane. Okay, so voltage gated and ligand gated channels, they're the two that you need to know about. The voltage gated ones are controlled by changes in ion concentration, and the ligand gated channels are controlled by the binding of signal molecules. Now, we've also got transport proteins, okay, transporter proteins, and what they do is they bind to a specific substance to be transported, and when they do that, they undergo a conformational change, which will then transfer the solute across the membrane. So in this circumstance here, okay, this is a particular type of transporter protein that we'll learn more about in a short while. But you can see what happens in this one, okay? You can see the conformation change in the protein there. So as a particular substance binds to the transporter protein, as we see here, it will cause a conformation change. And we'll talk about what's happening down here later on, okay? That conformational change of the protein then allows those molecules to be transported to the other side of the membrane, okay? So that conformation change is the key bit, and it allows specific substances to be transported by our transporter molecules across the membrane, okay? Those solutes can cross the membrane. Now, transporters alternate between two conformations, and again, we can see that in our GIF here, conformation 1 here, and in a moment we'll get conformation 2. Okay, so the two conformations alternate between each other. When it's in one conformation, it will bind with one substance. And when it's in the opposite conformation, it will have a high affinity or bind to another one. Okay, so active transport. Active transport uses pump proteins. Okay, pump proteins. Now, we came across those in the electron transport chain at higher, okay, where we had to pump the hydrogen ions against their gradient across the membrane, okay? And remember, it was the high energy electrons that gave those proteins the capacity to do that. So active transport uses pump proteins like this, and what they do is they transfer substances across the membrane against their concentration gradient. Okay, and we can see an example of those in the amoeba sisters GIFs there at the top. Now, we need a source of metabolic energy in order to carry out active transport. We know that from that 5. We need energy. Where does that energy come from? Well, it's our old energy currency that we learn about lots in biology, which is ATP. So we need some form of metabolic energy to pump these substances against the concentration gradient. Now, pumps that mediate active transport are usually transporter proteins, okay? Transporter proteins that are coupled to some sort of energy source. Now, some active transport proteins will hydrolyze ATP directly, okay? And we can see an example of that hydrolyzing reaction happening here, where we've got ATP, we break off that terminal phosphate, that third phosphate, and that is done by an enzyme called an ATPase, okay, or ATP hydrolase, but ATPase is fine for just now, okay? The ATPase carries out this hydrolysis of ATP, okay? Now, hydrolysis, and for those who are chemists out there, will know hydro water, using water to lyse, to split, to separate. So we're using water to separate that terminal phosphate from ATP. And we know that ATP stands for adenosine triphosphate, 3 phosphates. And it's been broken down into adenosine diphosphate. That's 2 plus this phosphate here. Now, we know that that phosphate, if it binds to a protein, will cause a conformation change, okay? And we'll talk about specifically how that does that in a few moments, okay? But the phosphate that's broken off, the energy we get from the breakdown of the ATP allows us to carry out this conformation change. And that's what allows us to transport or actively transport substances across the membrane against their concentration gradient. So, ion transport pumps and the generation of ion gradients. Now, this is where it gets a wee bit tricky, I think, in this topic. So, and we really need to focus on this part. So, for a solute that's carrying a net charge, okay, a positive or negative net charge, okay, a solute carrying the concentration gradient, okay, that's the number of those solutes or molecules, and so we'll underline that concentration gradient, and the electrical potential difference, that's the difference in charge on either side of the membrane. Okay, and our diagram here shows us this pretty nicely, I think. Those will combine, okay, so both the concentration gradient and the electrical potential difference will combine to form what we call the electrochemical gradient, okay? And that name kind of hints as to what it is, okay? It's the difference in the electrical potential difference, electro, and the concentration gradient, chemical, that determines the transport of the solute, okay? So it's this electrochemical gradient that will determine which way the solute will be transported across the membrane. Now, there is something called a membrane potential, and for those of you who have studied physics, you'll maybe have heard of a potential difference before. So an electrical potential difference. So that's the difference in the overall charge on either side of the membrane. So that is created, okay? The membrane potential is created when there is a difference in electrical charge on the two sides of the membrane. All right, so if one side is more positively charged than the other side, then we will get a difference there, which we call our membrane potential. Okay, the difference in overall charge on either side of the membrane. Now, there are things called ion pumps, okay? And as the name suggests, those pump ions across the membrane. And a good example of that is our sodium-potassium pump, which we're going to learn more about in a minute. But that uses energy from the hydrolysis of ATP that we learned about a moment ago to establish and maintain ion gradients, okay? And we can see it in this GIF again here, okay? So we're using ATP to cause a conformational change. How do we do that? Well, when we break off the terminal phosphate of ATP, it releases a phosphate, and that phosphate binds to the transporter protein. It causes a conformation change in the protein, and that then changes the affinity for the sodium. So it's a high affinity now, it's a low affinity with the conformation change. We now have a high affinity for the sodium, for the potassium. It will bind to the protein, the phosphate will be released, formation change will happen, and now we have a low affinity for the potassium and it's released into the cell. Okay. So don't worry about all those stages. We're going to go over them again in a second. But this is an example of an ion pump. It's called the sodium-potassium pump, and it's the one that's most often asked about in the exam. So it's well worth getting used to understanding this. Sodium-potassium pump transports ions against a steep concentration gradient. Okay. So it requires ATP because we're going against the gradient, active transport, okay? And what do we do to get the ATP? We have to hydrolyze ATP. So ATP hydrolysis is where we get our energy from. So this looks like a lot of steps here, but actually each step is very short in terms of the descriptors for it. So what we'll do is we'll use the diagram on the right here, and we'll talk our way through it using these descriptors that the SQA have provided on the left here. So initially, if we're at stage 1 here, at this first diagram, the pump has a high affinity for sodium, okay? High affinity for sodium ions inside the cell. And we can see on the diagram, sodium is binding here to its binding site on the inside of the cell membrane there, okay? Then, once the sodium has bound, okay, so binding occurs, we will phosphorylate the protein by adding a phosphate, this little phosphate here, which we get from the hydrolysis of ATP, okay? So we phosphorylate the protein from ATP using that phosphate from the ATP. What will that do? Well, when we add a phosphate, we know that this is true of all ligands. When we add a ligand to the protein, it's going to cause us a conformation change. That conformation change will change the affinity. Okay, initially we had a high affinity for sodium, but with the conformation change, the affinity for sodium will decrease. Okay, so you can see here that in stage 3, the protein has changed shape or conformation. At that point, the affinity for sodium is low, and therefore the binding sites let the sodium move out of the cell. So those 3 sodium ions there are going to move out of the cell. The sodium ions are released outside of the cell. What does this mean? Well, it means that we have 2 other binding sites here, which now have a high affinity for potassium. So they bind, the potassium ions there bind to their binding site. They've now got a high affinity. We then dephosphorylate the protein. That's where we remove the phosphate. Okay, we can see that happening there. The phosphate is removed. When we remove the phosphate, the conformation of the protein is going to return to its original state. Okay, so the conformation changes again, and then that will change the affinity for the potassium ions. The potassium ion affinity will become low, and potassium will be released into the cell. Okay, so those 2 potassium ions will be released into the cell, and our conformation and therefore our affinity for sodium will return to its initial state, which is where it has a high affinity for sodium from the inside of the cell. Okay, so that's the steps. It's well worth you memorizing them, but also, maybe sketching a wee diagram to go alongside these stages so you can visually understand what's happening. Because again, sometimes there might be a diagram or whatever in the exam that you have to refer to, and it'd be good to not only memorize, learn the stages, but also to know how they apply to the actual process visually. Now, for each ATP that's hydrolyzed, 3 sodium ions, okay, that's an important number to remember, are transported out of the cell, and 2 potassium ions are transported into the cell. Okay, 3 sodium out, 2 potassium in. You do need to remember those numbers. Why is the sodium-potassium pump so important? Well, it establishes both gradients, both concentration gradients and an electrical gradient. Okay, so both the concentration and electrical gradients within the cell. This is especially important in some of the other processes in the cell, such as one that we're just about to look at, but also another one which comes up in the next topic. So what are the functions of the sodium-potassium pump? Well, it's found in most animal cells, okay? So not just humans, but most animal cells. And it accounts for a high proportion of our basal metabolic rate. Okay, our basal metabolic rate is our base metabolism. It's our minimum required level of metabolism within our cells. Okay, for an adult human, it can vary, obviously. The basal metabolic rate can fluctuate up and down, but it's somewhere around about the 15, 16, 1700 mark, depending on the size of human that you are, and also on things like your physical activity and other things. So the basal metabolic rate, high proportion of just keeping us alive. That is what the basal metabolic rate is. It's the amount of energy we have to expend just to keep us functional. High proportion of that comes from the sodium-potassium pump, okay, and hydrolyzing all that ATP in order to do that. So this emphasizes the importance of the sodium-potassium pump, I think. Now, in our small intestine, okay, we utilize it there. Now, this sodium gradient that's made by actively transporting sodium out of the cell, okay, by driving that sodium, pumping that sodium outside the cell, that drives the active transport of another molecule called glucose. And that makes sense because we want to get the glucose that has been broken down and produced from starch in our small intestine, and we want to absorb that glucose into our cells. Now, quite often, the concentration of glucose in our cells is higher than it is within our small intestine. And so we have to actively transport that across, okay, from a low concentration in our intestine to a higher concentration across the membrane within our cells, okay? So what we do is we use a protein called glucose symport, okay? So the glucose transporter that's responsible for this is called glucose symport. Now that transports the sodium ions and the glucose ions at the same time. And we can see it in this diagram here, okay? Sodium has been transported through this protein at the same time as glucose is. Now, the difference is glucose is going from a low to a high concentration. Now, on its own, that would require a lot of ATP and energy to be transported. But because we're transporting it at the same time and in the same direction as this sodium ion here, which is going from a high concentration to a low concentration, i.e. passive transport, we can transport both. without having to hydrolyze all of our ATP in order to do that. So the sodium ions enter the cell down their concentration gradient. They're going from high to low. Meanwhile, there's a simultaneous transport of glucose, which has to be pumped into the cell against its concentration gradient. Okay, this one here is going from low to high. Okay, so from low to high, whereas our sodium here is going from high to low. And this transport happens simultaneously. That's why it's called a symport. Okay, simultaneously transporting the two molecules together. So in our intestinal epithelial cells, that's the cells which line our intestine, the sodium-potassium pump will generate a sodium ion gradient. It will make all the sodium go to this side so that we've got a gradient for it to move down, which will then simultaneously transport our glucose from a low concentration against the gradient to a high concentration inside our cells. Okay, folks, so I hope again that was a useful lesson. We've gone over membrane proteins there. Let me know if there was anything in particular you still are not sure on and you want me maybe to make another short video about, or any questions that you might have based on the content there, or just anything that you liked or disliked about the video. Until our next lesson, folks, thanks for now. Class dismissed.